Good afternoon, ladies and gentlemen. Welcome to the symposium on novel approaches to cancer therapy. First of all, I'd like to apologize to all, especially to our speakers. We never forget that we are still in the Middle East, and this was a short reminder. We are very honored to have the two laureates of the 2006 Dan David Prize in cancer therapy here. And I prepared the speech.
I'm aware of the fact that you didn't come here to hear me, so I'll be brief. Yet, you know what the difference is between politicians and professors. Politicians, when they see a mic, they must talk. Professors don't even need the mic. Now, with that in mind, I have a mic, and in 20 minutes, I'll greet you. First of all, I want to make 3 remarks. One regarding the disease, which some time ago was Yenemeise. Nobody even dared calling the cancer cancer. It became a chronic disease, or at least some cases, chronic disease. And it became a chronic disease through efforts and ways of treatment that had to do with either irradiation or cytotoxic drugs. I don't know what the proportion is of people that died of the drugs as compared to how many people died of the disease itself. It developed through ingenuity of many others to different lanes or different possibilities of treating. To the best of my understanding, most of the research that we are going to hear about today had to do with curiosity-driven research that didn't come with a recipe, as Feynman said, but are really breakthroughs. And Yitzhak, I really want to greet you for succeeding so well in nominating these 2 individuals to get the Dan David Prize. With that in mind, the only little remark that is not very optimistic has to do with the availability of the new technologies to the public. We are hearing stories about cancer of the gut, and the colon cancers and others that can be treated, or life can be prolonged, and people not getting those drugs just because of economical reasons. I'm not saying that we can expend 20% of our income, of our gross national product on drugs, but on health in general, but I'm quite certain that the fraction we are spending nowadays can be raised to accommodate for the new drugs and new technologies without too much of a national problem. Other than that, I'm looking forward to a wonderful couple of lectures. So is anybody else in the audience, I guess. So I'll give Yitzhak the mic back and wish us all very interesting 2 lectures.
Thank you very much, Professor Lichtenberg. I now call upon the Dean of the Faculty of Life Sciences, Professor Yoel Klug.
It is indeed a great pleasure, truly a great pleasure, to have 2 good friends, Professor John Mandelson and Professor Yossi Schlesinger, giving us first winning this prestigious prize, which they deserve, and I'm sure that this is only one way to the continuation of other prizes. It is a pleasure because it's rare that you can greet friends and colleagues that are in the field that you are working on for many years, and because I know Yossi and John and Yossi for many years, and I followed not only the outstanding research that brought them to drug development and drug into the clinics, but also many of the difficulties, and I can tell only few personal things. Once when I talked to Yossi many years ago, he said, when I said I'm going to isolate the EGF receptors, people said, you're not going to do that. That's right. And he did in big. And following this, there were the developments of the structure and the function and the signaling and the drugs, which eventually came into the clinic. John went a different way with the antibodies, and as I told Ann Mandelson today, this was a more difficult way to go through antibodies, but it was a long way. But eventually it happened. So I'm really happy to have you here, and particularly to have my colleagues and our students to listen to your talk. Welcome.
Thank you very much, Professor Klug. It's now an honor and a privilege to call upon Dr. John Mandelson, the president of the MD Anderson Cancer Center in Houston, to deliver his talk, Cancer Therapy, Targeting the EGF Receptor, Accomplishment and Challenges over 25 years. John, please.
Good afternoon, Professor Witz, Dean Lichtenberg, and Dean Klug, faculty and students and friends. Thank you very much for inviting me here. It's a pleasure to tell you a bit about an adventure that I've been on for the past 25 years, trying to develop a way to inhibit the EGF receptor tyrosine kinase. And there have been successes and challenges along the way. I'm going to go into the laboratory aspects as well as the clinical aspects, because many of you here that are PhDs, I know, are interested in how to get things into the clinic, and sometimes that's as hard as cloning the gene. Now, I have to begin with a disclosure. In the past, I have been a director of Mclone and held stock, and I do receive royalty payments from the University of California, which owns C225 or Erbitux, but I've never treated a patient with this agent, and I don't believe that it is ethical for someone who can receive income to do this. Now, in 1980, when I was at the University of California, San Diego, working with my colleague, Dr. Gordon Sato, we hypothesized that we might be able to produce monoclonal antibodies which bind to the EGF receptor and block the access of either EGF or TGF-alpha, and thereby prevent cell proliferation by inhibiting activation of the receptor tyrosine kinase. Monoclonal antibodies had been known at that time for 5 years. Nude mice had been known for 3 years. And as we'll see, there were only 2 other tyrosine kinases known at that time. So this was a novel hypothesis. Why did we make this hypothesis? A number of factors led to this. First of all, in 1980, Todaro and Sporn published their classic paper showing that tumor cells can produce EGF or TGF-alpha and auto-stimulate their own proliferation. At that time, there was a novel chemical activity, a tyrosine kinase, which was known to be the function of the Src oncogene and the EGF receptor. Stanley Cohen won the Nobel Prize in Medicine for demonstrating this and for purifying the receptor and the ligand. It was beginning to be known that EGF receptors are overexpressed by an order of sometimes 2 magnitudes in cancer cells compared to normal cells. There were experiments from the Salk and other institutions that for cancer cells, growth factors could be survival factors. That if you withdrew a growth factor from a malignant cell, sometimes these cells died. And importantly for a clinician, there were experiments of nature in which circulating antibodies, autoantibodies against receptors in patients, cause stable physiologic change, in this case, disease. So in myasthenia gravis, the antibody is against the acetylcholine receptor. In some forms of hyperthyroidism, the antibody is against the TSH receptor. And there are rare forms of diabetes where antibodies against the insulin receptor can cause change. So we made the hypothesis that we could produce an antibody which would actually block the binding of EGF or TGF-alpha. And we were able to achieve this. And these are the key publications in the early 1980s showing that blockade of the human EGF receptor by one of 2 different antibodies could inhibit proliferation of cultured cells and also cells growing in human tumor xenografts, and in collaboration with my colleague Gordon Gill, that the mechanism of action of this inhibition was blocking a tyrosine kinase activity in the receptor. So this was not immunotherapy, this was using an antibody as a tyrosine kinase inhibitor. Now, subsequent to that work, a great deal of research has gone on in academic institutions and also in companies today trying to block the receptor. Some work on the external side of the cell membrane. Don't see a pointer here. So the lateral line is the cell membrane, and above it is the external part of the receptor. And you can see that the antibody blocks the binding of ligand, and listed there are a number of antibodies that are in clinical trials. And then, logically, a large number of drug companies, including one that Dr. Schlesinger worked with very closely. Thank you, Isaac. Red button. Began to develop oral low molecular weight inhibitors called tyrosine kinase inhibitors, and a few of them are listed here that also block signal transduction. Now, when we began this work, it was with the hypothesis that we were blocking an autocrine pathway in 1980. Over the ensuing decade or so, we learned that there were many possible mechanisms where blocking the EGF receptor would potentiate cancer cells. Potentiate the treatment of cancer cells. This is a summary of the 6 capabilities of cancer cells published by Hanahan and Weinberg in Cell in the year 2000, a classic paper. There were 6 characteristics that cancer cells develop, and each of these 6 characteristics is potentiated and stimulated by stimulating the EGF receptor. Self-sufficiency in growth signals refers to the production of ligand. Insensitivity to anti-growth signals refers to the fact that these cells are not inhibited by TGF-beta and other sources. These cells evade cell death. Through a variety of mechanisms, apoptosis is prevented. There is limited replicative potential. The EGF receptor activates pathways that affect telomerase. Sustained angiogenesis, I'll show you some data, where the EGF receptor stimulates production of compounds like VEGF and basic fibroblast growth factor, which promote angiogenesis. And finally, activation of the receptor activates metalloproteinases and other molecules that potentiate metastasis. So this is, by total serendipity, we had an agent which in the right-hand column shows, and this has been shown experimentally in all of these cases except the telomerase, that blocking the EGF receptor reduces each of these characteristics of cancer cells. Now, there are other mechanisms of action of the inhibitors of EGF receptors. First of all, DNA repair after chemotherapy or radiotherapy is delayed and reduced. If it's an antibody mediating the attack, the antibody can carry out immunologic functions with these cells that express high levels of receptor. And it's been shown originally here at the Hebrew University, at the Weizmann, but subsequently in many, many laboratories, that chemotherapy and radiation therapy can augment the effect of an EGF receptor inhibitor. Now, let's look at some of the mechanistic studies that came out of our laboratory. Most cells are inhibited in G1 phase. We found that this is attributable to an increase in P27-KIP1 inhibitor of the cyclin-dependent kinases. These are immunoprecipitates from cells that have been incubated in the presence or absence of antibody 225 against the EGF receptor for 24 hours. Left-hand column is the control, and you can see here is the amount, the level of protein of P27-KIP1, and in cells that have incubated overnight in the presence of antibody blocking the EGF receptor, the amount of protein P27-KIP1 is markedly increased. The next two are enzymatic assays looking at P32 incorporation into a substrate histone. The cells are lysed and immunoprecipitated with an antibody either against cyclin E or against CDK2, and the precipitate is then looked at for its capacity to stimulate incorporation of P32 into histone. You can see strong incorporation here, completely blocked here. Now, we went through immunoprecipitates and tests with the other cyclin-dependent kinases and the other cyclins. These were the two that were most affected. And then looking at the RB protein in the control cells, most of it is in the hyper phosphorylated state, which would permit cells to go into S phase with release of transcription factors into the nucleus. And in the cells that have been incubated with antibody 24 hours, most of the RB protein is in the hypo phosphorylated state, which produces the G1 arrest. Now, similar studies have been done with all of the other molecules that have been produced against the EGF receptor, and they seem to all cause the same pattern for growth arrest. And Herceptin, the antibody against HER2, also activates increased amounts of P27 and blocks cells through the RB pathway. Now, this is an experiment carried out by colleagues at MD Anderson, a pancreas xenograft model in which the animals are sacrificed, and here we're looking at the control animals, and the red stain is for CD31. We're looking at the staining of new blood vessels, and the blue stain is FITC staining of dead cells with the terminal transferase reaction. When these animals were treated with gemcitabine, which is the best treatment, chemotherapy for human pancreatic cancer, when these xenograft-bearing animals were treated, you can see an increase in the number of dead cells, and you can see the endothelial cells here. In the upper right panel, you see an endothelial cell cut en face, the way you'd cut across a loaf of French bread. And you can see here the red stain are the endothelial cells on that blood vessel, and the green stain are the dead tumor cells, and the yellow stain is a superimposition of green and red, and these are dead endothelial cells. And when combined therapy of chemotherapy and antibody are given together, you can see a large number of dead endothelial cells and a large number of dead tumor cells. The mechanism of this was worked out in culture and in the animals. These tumor cells make large amounts of VEGF, basic fibroblast growth factor, and interleukin-8. And when the EGF receptor is inhibited, both in vivo and in vitro, the production of these angiogenic molecules is markedly decreased. And as I said before, in certain types of cells, and Dr. Josh Fiedler, my colleague at MD Anderson, feels that new vascular endothelial cells are among the cells that require VEGF or similar molecules for survival. And when they are deprived of these molecules, the cells die. Now, I mentioned importantly that the antibody can mediate immune effects. And this is an experiment done in collaboration with Ralph Reisfeld at the Scripps Clinic. And we're looking at melanoma target cells treated with normal human peripheral blood mononuclear cells. The melanoma cells are labeled in culture with chromium, and a chromium release assay is looking at cytotoxicity. And the hatched bars here show the amount of chromium release in 4 hours when various concentrations of antibody are added to these cultures. And this is the human chimeric antibody. That's what C225 is, that I'll describe in a little more detail. The control is the same cultures treated with the mouse 225 antibody. And of course, the mouse antibody would not be recognized by the human ADCC cells, and there is no cytotoxicity with mouse 225, and there's prominent cytotoxicity with the human chimeric version that these cells can visualize or can understand. There are other studies that have been done to support the immune effects of this antibody with a lecture that I was told to keep to half an hour. I won't go into them in more detail now. Now, this is one of many studies in the xenograph combining an EGF receptor inhibitor and chemotherapy. And this happens to have been done with Dr. Ciardiello in Naples. I've had many collaborators in many countries, actually. And you can see here the control growth of the xenographs. And here the animals were treated with topotecan, which is one of the best drugs against colon cancer. These are colon cancer xenografts. Here the animals were treated with antibody 225. And here's the combined therapy. The treatment lasted 1 month, and you can see that eventually, in the majority, not all of the animals, the tumors did grow out, but there was a marked improvement in the response when there was combined therapy given. Now, this kind of approach has been done in our laboratory and with collaborators with a wide variety of tumors, and it's been done with each of the EGF receptor inhibitors and other types of kinase inhibitors and signal transduction pathway inhibitors that have become truly an industry over the past 20 years. Now, where can a person running a laboratory in an academic institution go if they would like to take this commercial? There's 2 ways to go. One is to start a company, and my wife, who's sitting in the second row, did encourage me to do that. But instead, I said no and talked to the University of California, which had the patent, and it was licensed to the first of 5 companies that I've worked with in the past 20 years. This was Hybritech. It was the first biotech company in La Jolla, which now has 150. This was number 1. They were very interested in this antibody, and after licensing it, they spent, I estimate, about $5 to $10 million in scale-up and formulation of a mouse monoclonal antibody, carried out the toxicology studies, and worked with the FDA. And in 1989, a phase 1 trial was carried out at Sloan Kettering, which is 5 years after the patent had been applied for, and nearly 10 years after the original idea. Now, what was learned in the phase 1 trial? This was the first clinical trial in a human being with a receptor inhibitor, and it was the first clinical trial in a human being with a tyrosine kinase inhibitor. And massive toxicity was the worry of ourselves, the FDA, and the patients. Fortunately, there was only modest toxicity. We gave a single dose per patient up to 300 milligrams. Why did we stop at 300 milligrams? The company ran out of antibody. If you try to produce antibody and ascites and do clinical trials with it, that is the rate-limiting factor. At doses more than 4 grams, the antibody imaged the tumors. The antibody was tagged with indium-111, and it imaged all the tumors in the patients with lung cancer that were studied and all of their metastases that were larger than 1 centimeter on a CT scan. So we showed that the therapy reached the tumor and the metastases. At concentrations higher than 120 milligrams, the serum levels were maintained at higher than 40 microgram per milliliter for at least 3 days, which is enough to saturate receptors, which we had shown in all of our preclinical work was necessary. And within 2 weeks, each of these patients had made human antibodies against the murine monoclonal 225 antibody. And that was published in the JNCI in 2001. Now, at this point, that was a study with a murine antibody. At this point, the NCI was convinced that this would be an exciting approach to cancer therapy and paid for the production of a human chimeric version of murine 225. It's a human IgG1, which does bind complement and mediates ADCC. It has a higher affinity for the receptor than EGF itself, and it does compete with the growth factor for binding, inhibits the kinase, and stimulates receptor. Now, you'd think that we could move quickly from that point, but that didn't happen. Why? There was a hiatus of 4 years. Further clinical trials were carried out. First of all, there was general skepticism about antibodies as treatment for cancer. It's hard to remember that 15 years later, when half the new drugs against cancer are antibodies, that as late as 1990, no drug company had ever produced an antibody that helped a patient or made any money for the drug company. They're high molecular weight. It was felt that they would have poor distribution. They must be given intravenously. They're very hard to produce and expensive. C225 is produced in huge bioreactors, not in mouse ascites, so you can get large quantities, kilograms out of 100 and 1,000 liter bioreactors, but they're very expensive. There's always the potential for an allergic reaction, and as I said, there's no track record. And in the meantime, once this mechanism had been shown to be effective in vivo model systems and safe in patients, there was tremendous interest to develop oral agents. And most importantly, Hybridtech was bought by Eli Lilly, which had had a bad experience with the previous antibody, and essentially nothing happened for 4 years. Now, Mclone was a company in New York that learned about this work and was very interested in obtaining the license. And fortunately, the University of California was able to get the license back from Lilly without a lot of legal hassle. I think it would be harder today. And Mclone, in turn, licensed C225 from the University of California, built bioreactors, first subcontracted, and produced huge quantities of this agent, carried out formulation and toxicology studies, and began working with the FDA. Sponsored phase 1 and 2 trials, which included pharmacokinetics. And among these trials was the first that demonstrated in a human being that a receptor kinase inhibitor could reverse resistance to chemotherapy in patients. This was at MD Anderson in patients with head and neck cancer who had been treated with cisplatinum and had failed therapy, and they were treated with platinum plus the C225 and had responses, which had not ever been seen before as an approach to therapy. The pivotal trial was designed for accelerated FDA approval, adding C225 for advanced colorectal cancer in patients who were progressing on the best drug against colorectal cancer at that time, which is irinotecan. Now, these are the results of that study, which were reported in 2001, about 15 years after the original work was hypothesized and 13 years after the first publication. This was carried out at Sloan Kettering and at Yale and a number of other schools, and the lead author, Dr. Saltz, showed data that in patients who were progressing on irinotecan therapy and then were treated with irinotecan in combination with C225, 121 patients, the response rate was 22.5%. 22.5% of these patients now on irinotecan with the addition of 225 had an objective response. There was also a group of a quarter of the patients whose disease stabilized, and the median duration of the response was about half a year. The data shown here were not available at that time. This is a study on identical patients carried out with just antibody. Incidentally, C225, the chemical name is cetuximab and the trade name is Erbitux, and I often use them in a mixed-up way, but they all are the same molecule. The response rate to antibody alone in a similar group of patients is only 10%, stable disease about a third, and the median duration of the stable disease also is substantial. But these data were not available to the FDA. These were the data that were available, and this is what happened. The FDA turned down the trial, and this is a famous event, not because of anything to do with Erbitux, but because Martha Stewart had some problems related to allegedly knowing in advance that the FDA was going to turn this down and selling stock. Why did the FDA turn down, and it's something I'm sure you've read about in the newspapers. You probably haven't read much about Erbitux, but you've read about Martha Stewart. That's the way our world works. Why did the FDA turn down this trial? They claimed that the data records were incomplete, but that could have been fixed, and they claimed that the x-rays demonstrating progression on irinotecan alone didn't adequately document. But the main reason why the FDA turned this down was there was a requirement to prove that C225 alone couldn't achieve the same level of response in the absence of chemotherapy in a randomized trial design. And many patients were unhappy about this, and however, as you will see, a company that was collaborating with Mclone, Merck, and now we're with a second drug company. The first was Lilly, the second is Merck, and I didn't mention, but on the previous slide, Bristol Myers Squibb was mentioned as a collaborator with Mclone. Merck Germany did carry out the randomized trial. They had started it prior to the FDA ruling. It was reported in 2004. This is taking patients that are progressing on irinotecan. Two-thirds of the patients received irinotecan plus C225, and one-third of the patients received C225 alone. And you can see the response rate was very similar to the previous trial, 23%. The combination of response and stabilization of disease, about half the patients' time to progression was 4 months, and the median survival time was 8 months. Now, in these patients that had the monotherapy with antibody alone, the response rate was very similar to the previous trial, 10%, 32% stable disease. The time to progression was very short, and these patients were allowed to cross over and switch to this. So most of these patients received the combination therapy once the disease progressed. So these two numbers can't be compared. Looking at these data, the FDA went ahead and approved Erbitux for the treatment of advanced colon cancer, either with combination chemotherapy or alone, based on these response rates. Now, this approach to cancer therapy has been studied with a wide variety of tumors. The most recent FDA approval was in March of 2006 for stage 3 head and neck cancer that could not be treated with surgery. The primary therapy for this is radiation therapy. And here was a randomized trial where half the patients received radiation therapy for cancer in the throat, in the tongue, in the neck, and the other half of the patients received radiation in combination with cetuximab. And these are typical curves that look very close, but if you do look at the median survival, you can see that the median survival for radiation alone was 28 months, and the median survival for the combination therapy was almost twice that. And if you follow these patients out for 3 years, the disease-free survival rate is about 10% higher in the combination treatment than the control. And on the basis of these data, the FDA has approved Erbitux or cetuximab for the treatment of head and neck cancer. There are active trials that I'll mention in a minute going on with a variety of other types of cancer. So the summary of the clinical data to date, nearly a dozen different competing anti-EGF receptor agents are in clinical trials. They compete with EGF or TGF-alpha for the receptor, and they also compete with each other. And they're against a wide variety of cancer types. The response rates for single-agent therapy vary from about 0 to 13%, with the exception of patients with non-small cell lung cancer in Asia, where the response rate is higher, and they vary with the agent and with the tumor type. And a drug called gefitinib, which is an oral tyrosine kinase inhibitor, and another drug called erlotinib failed to show enhancement of survival in these lung cancer patients when they were added to chemotherapy in large randomized clinical trials. Now, these were huge clinical trials involving thousands of patients, and it was expected that combining these oral agents with chemotherapy would improve survival compared with chemotherapy alone, and 4 different clinical trials were negative. So you can see that lab data doesn't always predict clinical data, because they had lab data similar to what I showed you with Erbitux. Gefitinib was the first tyrosine kinase inhibitor against the EGF receptor approved by the FDA for lung cancer. The response rate was 10%. A subsequent randomized trial did not improve survival, and the company has withdrawn gefitinib for lung cancer. The second to be approved was cetuximab or Erbitux, and I've already gone over those data. The third was Tarceva or erlotinib, approved by the FDA for refractory lung cancer, with response rates of 9%. And I mentioned the C225 or Erbitux is also approved for head and neck cancer. Now, let's talk about the differences between antibody and tyrosine kinase inhibitors. They are different. In colon cancer, you saw that 10% of patients respond to the antibody alone. There's never been a patient that responded to the various oral tyrosine kinase inhibitors, blocking allegedly the same pathway. So these antibodies act differently with different patients. With the oral agents, gastrointestinal toxicity is rate limiting. Intolerable diarrhea. In the case of antibody, blocking the same pathway but with a different mechanism, there is no GI toxicity. All of these agents cause a skin rash, as we'll get to in a minute. The antibody augments chemotherapy and radiation therapy in patients. I've shown you the data. Up till now, the oral tyrosine kinase inhibitors do not do this. The antibody does mediate antibody-dependent cytotoxicity, and of course the oral agents can't do this, and the antibody downregulates the EGF receptor, and the oral agents do not. Somewhere on that list, with more probing, we're going to learn about the differences between these agents. Now, there is one toxicity which actually predicts response, and that is a skin rash that looks like acne. It's strange to see acne on adults and older patients all over the face and the chest, and it can spread over the body. In the original study reported by Dr. Saltz, 27 patients responded to therapy. 26 of those patients had a skin rash. Only one patient that lacked a skin rash had a response to therapy. Highly statistically significant difference, and this has been borne out in most of the studies with the various inhibitors. Now, there's a related antibody called Herceptin that I'm sure you're aware of that blocks HER2. This was developed by Genentech subsequent to the publications that we published in the early 1980s. And in that case, the level of expression of the HER2 does predict response. And in first-line therapy, if there is a 3+ level of expression of HER2, 35% of patients respond, and if there's less, no patients respond. We had felt that it was likely that this would be the case with EGF receptors, but you can see the data from Dr. Saltz's study, confirmed in numerous other studies with our antibody and with other agents, that 1+, 2+, or 3+ expression of EGF receptor didn't matter. It didn't predict the response rate, and the dependence on this pathway enough to have clinical relevance did not get predicted by the level of receptor expression. Now, this is a study that was done on skin biopsies by my colleague Dr. Bazalga and ourselves, looking at patients treated with C225, and here we're looking at MAP kinase. The dark stain is phosphorylated MAP kinase in the base of the skin before treatment, and after treatment you can see that is eliminated. Here we're looking at Ki67, which is a surrogate marker of S phase and cell proliferation, prominent in the base of the skin and absent, or nearly absent, after treatment. There have been many studies of this type done recently with biopsies of tumors, and we expect to have data which will help us predict who is responding and who is not responding in the near future. But those data are not available, and it's sad to say that I estimate that over 100,000 patients were treated with various approaches to blocking the EGF receptor before any serious look to try to predict who would be a responder and who would not was begun by the various companies involved. Now, there are mutations that have been found in the EGF receptor only in lung cancer, with very rare exceptions, and they're important because we've learned a lot about sensitivity and resistance to drugs. They're not relevant to the antibody approach. So these mutations are seen only in lung adenocarcinoma, oddly enough in non-smokers, females more than males, and Asians much more than Americans. And some of these mutations especially a missense at the locus cited there, and in-frame deletions in the locus cited there. If those mutations are present, that's found in 80% of the people that respond to the oral tyrosine kinase inhibitors and only 6% of non-responders. And overall, the response to these inhibitors in the United States is about 10% with an average duration of 6 months. There's a few patients with prolonged responses. And I should say that there are some patients I know that have been on Erbitux for 4 years. There is no way to predict this subgroup of patients that have prolonged responses to tyrosine kinase inhibitors. These mutations in lung cancer are important because benefit does occur to patients that have these in terms of partial remissions and in terms of long-term maintenance of stable disease. The mutated receptors are more active. They have a higher degree of autophosphorylation. Cells with mutated receptors are more sensitive to chemotherapy, and treated patients live longer. And the treated patients with chemotherapy live longer whether or not they received the tyrosine kinase inhibitor. So there are some people that feel that this form of the mutated EGF receptor is present in cancers which have a less indolent course. And these cancers are in patients who live longer with or without treatment of the tyrosine kinase inhibitor. Now this gets very complicated to sort out from retrospective clinical trials, and it'll have to be looked at prospectively. It's been shown in a number of laboratories that resistance develops to the oral tyrosine kinase inhibitors in these patients with mutations due to additional mutations which have been shown with structural analysis to actually block the binding of the tyrosine kinase inhibitor. So that kind of study was done within a year of finding resistance, and it shows you how the technology has progressed, that we can actually now identify mutations that cause drug resistance and show that it blocks the binding of the drug to the site on the ATP binding site. Mutations also occur in RAS in some of these patients, and that precludes a response to the tyrosine kinase inhibitors. More recently, a number of laboratories have been looking at the copy number of the EGF receptor, and in patients now treated with one of the oral agents, a study that came from Denver, you can see that if the copy number is increased, 33% had an increased copy number, the response rate is well over 30%, and if there's not an increased copy number, there's a very low response rate, highly significant. The median survival, quite different, and the 1-year survival quite different. In that same study, looking at mutated receptors, there was also a difference, but the 1-year survival was not statistically different. But if you compare those numbers and those numbers, they're not that different, and clearly we need a larger study. Now, how does this apply to the antibody? These are data, again, with the oral tyrosine kinase inhibitors. In the case of the antibody against the EGF receptor, the EGF receptor mutation is not relevant. The antibody works whether or not there's a mutation present in 10% of patients. The gene copy number is being studied. The level of phosphorylation of the EGF receptor prior to therapy is being looked at. The downstream pathways that I'm sure you'll hear more about in just 2 or 3 minutes are being examined in biopsies from patients, and there's evidence that possibly the level of phosphorylation of AKT or MAP kinase or the level of P10 or the activity of the PI3 kinase pathway all may be relevant to predict those who might respond to blocking the EGF receptor with cetuximab. We're looking at the ligands present in the tissues and at the presence of the paired receptors, HER2, 3, and 4, along with HER1 or B1. So in conclusion, the hypothesis that inhibiting the activation of the EGF receptor tyrosine kinase can be a useful therapeutic approach to cancer is correct. Antibody 225 against the EGF receptor is an effective anti-cancer agent for some patients. And clearly we have a great deal more to learn in order to identify the tumors that depend on this signaling pathway and to optimize treatment with cetuximab either alone or in combination with other therapies. So at the present time, there are over 57 clinical trials going on that I know of, and this, of course, is one of the things that raises the costs of these drugs, all the trials that have to be done. With chemotherapy, C225 is being looked at with a wide variety of tumors. The most aggressive studies are up front in colon cancer as adjuvant therapy or as first-line therapy, rather than looking at patients with advanced therapy that have failed chemotherapy. Also, many types of cancer are being studied with radiotherapy. Everyone in this room is aware that there are EGF receptors present on every epithelial and mesothelial cell in our body, so theoretically any of these cells could be targets. There are a number of novel approaches being taken that all depend on preclinical data saying that they might work, combining C225 with antibody against the VEGF receptor, with antibody against HER2, with another agent against the EGF receptor, and with a blocker of COX-2. The xenograft data all suggest that this may improve response. And all of these studies now, as I mentioned, are including pharmacodynamic studies. That means biopsies are being done, and we're looking at the serum. Now, the other conclusion drawn from this work is that the research took place in 3 universities. It involved collaborations on my part with dozens of laboratories and the National Cancer Institute, which, after all, funded about $20 million worth of research and paid for the chimerization. The commercialization involved 2 biotech companies and 3 pharmaceutical companies over a period of 22 years. Clearly, there's a need for markers that would predict sensitivity. It's understood now. It was not appreciated as far back as recent as 1990. I believe there's a need for new FDA guidelines on studies that involve some of these therapies and combinations, because many of these new agents are only going to work in a small percentage of patients, but in combination they're going to work much better. And as long as you have to prove efficacy with an agent alone before you can combine it, we're working with one hand tied behind our back. And I guess the most important lesson that I've learned from this is perseverance, accept setbacks, and be lucky. Thank you very much for your attention. There's time for questions. I'd be glad to take them.
I think we should move on. Okay. Thank you very much, John, for a wonderful lecture. We have to go on. Dr. Schlesinger, as well as Dr. Mandelson, have to work still today, so we'll have to give them some rest. Can you please sit down and be quiet? Thank you. It's now my pleasure to call upon Professor Yossi Schlesinger from the head of the Department of Pharmacology at Yale, who will talk about cell signaling by receptor tyrosine kinases, from basic concepts to cancer therapy. Yossi, please.
Where's the laser? Where's the laser? For pointer. Okay, yeah. Okay. Many thanks for all of you, for the prize committee, for the deans on coming here, and for all of you coming to participate in this lecture. Since I'm a basic scientist, I'm going to entertain you in this afternoon. It's unavoidable to hear some new scientific concepts about how receptors are activated. So basically, I'm going to tell you several stories. The first is, I'll give you a brief summary of the fundamental concepts, which led also to the work that John described. I'm going to tell you a very recent result, which is rather surprising, which shows that receptor tyrosine kinases are activated by well-ordered reaction, which allows us to indicate, to suggest that there is a two-state activation of receptor tyrosine kinase. This is rather new and provocative result. I'm going to show you the discovery of a tyrosine kinase inhibitor as a novel cancer drug that was approved early part of this year and is extremely efficacious for certain cancer. And finally, I'm going to show you a new strategy that we developed for a robust production of a variety of drugs, which I believe will change many aspects of that field in years to come. Just a brief introduction. I've compiled here for some people who are interested an historical perspective of this field, and I've ended historical aspect in 1990 because there was an explosion in 1990, and I don't want to make more enemies that I already have by not quoting some people's. But in the early days, it was rather clear who did what. So I'm going to tell you what happened and what led to what from 1960s to 1970, although I'm not that old. So in the 60s, EGF and FGF were discovered, and this is not just EGF, this is the entire field of receptors, tyrosine kinases, and their signaling. This is what has happened and led to all. 70s was an interesting time, although the pace at that time, as you're going to see, was much slower. So in the 70s, here was a seminal paper who changed my interest in this, because I was, as many of you know, I was a biophysicist, spectroscopist who were trained in biophysical studies. I read this paper by Howard Hammond that showed that cancer cells need less growth factors for proliferation, and cancer cells produce their own growth factors. So this was an interesting study which really led me to believe that this could be an interesting target to explore. At that time, it was also shown that insulin and EGF bind to cell surface receptors. In 1978, we have shown, and some other labs have shown, that EGF and insulin receptor undergo receptor-mediated endocytosis. At that time, it was thought that maybe the interactions are dynamic on the cell surface. And in 1979, I proposed the fundamental concept, that dimerization model for EGF receptor, which turned out to be a universal mechanism for the activity of nearly all surface receptors which have a single transmembrane domain. In 1980, as John already mentioned, SARC was shown to be protein tyrosine kinase, and shortly thereafter, EGF receptor has been shown to be protein tyrosine kinase activity. And in 1983, there was an interesting development which showed for the first time that a cellular oncogene encodes for a growth factor. This was shown to be the PDGF receptor. So this was the first mechanism which identified a molecule which is an oncogenic protein. And this year we were the first to purify the EGF receptor. In 1984, with Mike Waterfield, we have shown that VRB oncogene is derived from an EGF receptor. This was the first demonstration that a receptor tyrosine kinase is an oncogenic protein, which provided the conceptual foundation for everything that John reminded in terms of the link in cancer and how to develop it. EGF receptor was cloned in an effort with Axel Ulrich, and EGF receptor was shown to be amplified in brain tumors. This is work that I did here with a graduate student at the Weizmann Institute when I was still a faculty there. Subsequently, CFEMs was shown to be oncogene and family of receptors create a consistent picture, and this is the family of FGF receptors. The human genome contains 22 FGFs, which are depicted here. This is an interesting, huge family of growth factors which induce a great deal of responses. They mediate the responses by activating a FGF receptor, and here I have an interesting story where a single gene can encode for 2 different receptors through this following mechanism. So, for example, for each of these receptors, there are 4 of them, you have the following arrangement. The extracellular domain is composed of 3 IgG-type domains. This is a major determinant of ligand binding specificity. It's encoded by 2 exons, exon 7, which is common to both of them, and exon 8, which is common to the B form, and exon 9, which is common to the 9 form. Now, it turned out to be that the B form binds a selective group of ligand. This form of the receptor is exclusively expressed in the epithelium, and all the ligands are expressed in the mesenchyme. This is really different for the C form, which is expressed exclusively in the mesenchyme, and all the ligands are expressed in the epithelium. So this is a very interesting mechanism for epithelium-mesenchyme interaction, and this program goes wrong in some cancers. So you can see how a combination of such a ligand would lead to cancer or other diseases. Now we have established the mechanism of this and many receptor tyrosine kinase, but in this particular case, this is sort of the mechanism of how the kinase receptor is maintained in an inactive state. This is the ligand binding domain, it's excluded, and this also receptor binds heparin, so this is all blocked in the inactive state. Once the receptor is activated, and so here we used structural approaches to determine receptor function. So here you can see the structure of the extracellular ligand binding domain. This is the region where the ligand binds. So you see this is a ternary complex composed of 2 heparin molecules, 2 ligand molecules, 2 receptors. This is a real structure. This is not a model. We would like to obtain the structure of the intact molecule, which is one of the projects in the lab. In the meantime, we have also determined the crystal structure of the catalytic kinase domain. This is the kinase that John was describing in detail, and we are interested in understanding how molecules block this activity. It has all the hallmarks of a protein kinase, nucleotide binding site, and these 2 tyrosine residues must be phosphorylated for the kinase to be maintained in an active state. So the first event which happens to all protein tyrosine kinases is that they become autophosphorylated. And some of these autophosphorylation sites serve not only as a signaling for activation, but also as a vehicle for recruitment of signaling molecule. And this is the first subject which I want to going to tell you, which is an interesting new story about how receptors are being activated. So this is the following story. So this is how one of these receptors looks like. Extracellular domain and the cytoplasmic domain. So these are the multiple tyrosine phosphorylation sites. These 2 sites are in the activation loop. These 2 sites are in the activation loop. They regulate the tyrosine kinase activity. So we produce large quantities of this protein tyrosine kinase for structural studies, and these are the phosphorylation sites which are phosphorylated. And then we ask a very simple question, which looks really simple and useless, and which, as you will see, the consequences are rather interesting. What is the order of the phosphorylation of this reaction? And when you have 5 phosphorylation sites in a kinase, this is the combinations to phosphorylate the first site, second site, the third site. So you see there are many, many combinations. So you have a tyrosine kinase that becomes phosphorylated. Is it really an ordered reaction? If it's an ordered reaction, what does it mean? If it's not an ordered reaction, is it really random? If it's really random, what does it mean? Well, let's first see whether it's ordered or whether it's random. So the way we studied it, this is the sort of state-of-the-art approach to study this process. You take the tyrosine kinase, you add ATP and magnesium, and then you mix them and you analyze using mass spec the different phosphorylation state, and you really purify them also in run and gel and to see the different phosphorylation states. And this is how it looks like. So as you can see, the reaction turns out to be dramatically ordered. So this will be non-phosphorylated kinase, the first site, second site, third site, fourth site. It's really like you speak about physics. It's like a quantum steps. Every site that is phosphorylated, this kinase move. So you can really draw kinetic curves and establish, and I'm sure that most of you, if perhaps not interested, just to show you that this can be done. And so this is measuring all the kinetic steps, and you can really draw a detailed different steps of the kinase to be phosphorylated, the first site, and also all the kinetic values can be derived. And this is the kinetic mechanism. I'm not going to dwell on that. This is for the expert. But basically, we can see very clearly that the reaction is ordered, and you can really determine all the kinetic parameters. But here comes the question. What is the order of the reaction? And the answer turned out to be rather interesting. So when I show it to people and I have more time, I make a quiz. What do you think would be the order? I'll show you what the order is. The first site that is phosphorylated is tyrosine 653, which is here, first site in the activation loop. Well, we always thought that the second site in the activation loop will be the second, and we even predicted that. This turned out to be totally wrong. So the order of the reaction turned out to be the following here. This is the second site, and this is the number, and this is the position here. The third site, the fourth site, and the fifth site. The second site in the activation loop is the fifth site. So this really has a tremendous implication because it shows us how receptor tyrosine kinase is activated and how the kinase activity is regulated vis-a-vis two-step model. So in order to really characterize this in detail, we have isolated a kinase which has one phosphorylation site in the activation loop, purified it, a kinase which has two phosphorylated, and measured the activity of this kinase to phosphorylate an substrate. In this case, it's phospholipase C gamma. So here you can see that the kinase which is not phosphorylated is a weak enzyme. Once you add one phosphate, there's a 54-fold increase in the kinetic properties. Once you add another phosphate, it's about 500. So this has tremendous implication. It shows you that there is two-step activation. So the reaction which John was describing is a very ordered reaction. It tells you something rather insightful about how the steps of this enzyme activated. Also, it will tell you how to inhibit it in a very sort of regulated fashion. So this will be the conclusion or this will be the summary of this part of the lecture, that basically what we have now is a two-state for activation of FGF receptors. We believe that this is rather universal. The first step is shown here, that the first site is activated. This leads to about 50 to 100-fold increased activation. The second site, then at this stage already, autophosphorylation can take place, which leads to recruitment signaling molecules. So signaling molecules can already be recruited before the kinase is fully active. So the first step already leads to recruitment. So there is an assembly which is concomitant with just partial activation of the kinase. Then the kinase becomes really super activated, which leads to the second step. Now this is a fully activated kinase, which then phosphorylates this exogenous substrate. So this is based on that we propose the mechanism of two-state activation of receptor tyrosine kinases. Now, what is the mechanism of this process? This is such a delicate reaction, it was rather difficult to establish the mechanism. So I'll just show you very briefly some recent unpublished results. I'll just skip this because I showed you the scheme. So basically, the way we determine the mechanism, since all phosphorylation occurs in trans, namely one receptor can phosphorylate the second one, we created a battery of receptors which contain only one phosphorylation site at a time, and then use wild-type receptor to phosphorylate them. Then we measure the kinetics of each. So this way we can really establish the rate of each receptor phosphorylation. And using these panels of all phosphorylation sites, we were surprised to find out by measuring the mobility in gels, which is a measure of the phosphorylation, that all these events are kinetically controlled. Basically, this is a kinetically controlled reaction. The first site is phosphorylated, the kinase is activated, then the rest is determined. We still don't understand. We think this is very significant, but we know that this process is kinetically controlled. So this shows you these phosphorylation sites, where they are located. We still don't understand why all these sites, which are rather similar in some aspects, are kinetically controlled. There are significant differences. This is something to explore in the future. So I wanted to show you one basic science question which we're addressing, because I wanted you to realize that without this understanding the concept, we will not be where we are in terms. So the major interest of the lab is deciphering the mode of action of how this molecule works. In addition to that, it became clear to me in the 70s that this is a useful approach to drugs. So my founding Sujan, it's a company we founded in 1991 as the first company whose goal was to develop new drugs for the treatment of cancer and other diseases caused by dysfunction in protein tyrosine kinases, protein phosphatases, and their intracellular signaling pathway. It was rather difficult to convince at that time, but this was a boom area in the time of biotechnology, and it was not so difficult to raise quite a lot of money. These are the different people that have been participating in this company, and I color code them. Eventually, the company had 450 people. So these are the three founders of the company, myself, Axel Ulrich, and Stephen Evansvik, who was the CEO, and you can find some people here, some local people as well, who spent some time in the company. So this is an exercise now in American capitalism, and John showed you an earlier exercise. So Sujan was founded in 1991. Sujan was acquired by Pharmacia in 1999. Pharmacia merged with Monsanto in 2000. Pharmacia was acquired by Pfizer in 2003. And the drugs that were developed by Sujan have been developed now by Pfizer. All the drugs have been derived the name SU for the founders, Axel Ulrich and myself, although I just realized last night that my son, Schlesinger Udi, thinks that I named the drugs on his name. So this is the family of drugs that have been developed. This is eventually the drug that became successful in the clinic. And this is some kind of derivative of this drug that really clarified to us how this family of drugs work, because we determined the crystal structure of how these molecules, which tell us how these molecules act. So here you see the crystal structure of the one of these indolinol derivatives in the pocket of ATP binding site of FGF receptor. We have also other crystals. You can see this can be superimposed with ATP, so this will be the compound, and this will be ATP, so they really compete for the binding of ATP. So this is a slide which I got from George Dimitri from Dana-Farber, which shows you as a single orally administered agent how efficacious it is. The efficacy is about 40% of patients. This is results on GIST, or gastrointestinal stromal tumors, which are also resistant to Gleevec. So this is fully metastatic patients here. This is a scan, and this is after 7 days of treatment, single agent, orally administered. So all this, these are all normal organs. So the response is really rather remarkable, very similar to Gleevec. So since the response was so efficacious, it was granted a fast-track approval, and in February 8, this was given, this fast-track approval, and since the effect was so dramatic on about 400 patients, it was immoral to continue placebo to the 110 patients, and they were terminated, and the clinical trial was stopped because it was really rather efficacious. So this is eventually how this drug looks like. So it received FDA approval on January 26, 2006 for the treatment of renal cancers, gastrointestinal stromal tumors, and for Gleevec-resistant GIST. It shows promising results also for the treatment of other cancers, which I'm going to show you. It blocks the kinase activities of PDGF receptor, KIT, and VEGF receptor, and it acts in different cancer in different combinations. This is why it's so efficacious. This is an article which appeared in Business Week showing how efficacious it was to renal cancer, and this is one of the patients which was very sick, which really showed a tremendous response. And this is in June 13, 2005, just about a year ago. So this is the approval letter by the FDA showing this approval. So it took about $400 million and about 15 years to develop this drug. So this is the effect of this gas on breast cancer, also as a single agent. it was a surprising result since we still don't understand the basis for that. It has a significant single-agent activity in patients with refractory metastatic breast cancers. 16% response was seen with 51 patients of heavily pretreated patients. Toxicity is manageable and rarely limiting therapy. The current focus is on less heavily pretreated patients. I believe that there are going to be some announcements about this and on melanoma showing that this drug is rather efficacious. So the question is, if this is so good, why only so few scaffold chemical entities for cancer therapy have developed? So this was a slide I prepared 5 years ago. So at that time, these were the only examples of non-tyrosine kinase inhibitors that were considered for clinic. The family of quinazolones, purines, oxyindoles, and pyridine. This was our contribution. This is a Sujan work. And look how many patents. So the question was why were so few families of protein tyrosine kinase discovered? So at that time, Sujan was already part of Pharmacia, and I tried to convince them about a new approach to develop new families of inhibitors. But they were really rather rich at that time. They had really integrated in Pfizer. They didn't have the incentive to start something daring. So I decided to start another company, and in order not to have any discussion about priorities, I also made myself the chairman of the board of the company. So Plexicol was founded in December 2000 by Sanho Kim, a crystallographer, and Peter Herr. He was a former president of Sujan, and myself as the first company whose goal was to employ scaffold-based approach and co-crystallography analysis of drug protein tyrosine kinase, drug protein complexes for drug discovery. The most advanced programs are in the fields of diabetes and metabolic disease, nuclear hormone receptors in cancer and rheumatoid arthritis, and in inflammation. I'm going to show you one example which I think is going to be a very successful molecule in cancer. So first of all, what is the approach? Well, I've thought why all of us are really failing to discover new chemical entities. And I've done an experiment in my head, and that is, I said, why do we all fail? So I reached the following conclusion, and it's always nice to do it in your head because you always succeed. I said, we all get this few inhibitors because we are all using a very similar assay. We are all using similar databases, and all of us are looking for champions. And repeatedly, we were finding the same molecule. So I said, well, instead of that, instead of looking for champions, let's look for the wimps. Let's look for those which are weak, and then try to build up specificity and selectivity through building affinity through structure. So since my investors were very happy because they made a lot of money with the first company, it was rather not so difficult to raise about another $60 million and to start the following approach. So this is what we engineered. So this is a real experiment. So you take 3 protein kinases and you look for common inhibitors. So this would be common inhibitors to 3 protein kinases. Look how many hits. And you look for really weak inhibitors. No one cares for them. They were all thrown away by the Pfizers and the Mercks. These are very weak inhibitors. Now, we think that we know how to handle them only after we will see where they bind. So the way we solve the problem is we do high throughput co-crystallography. This was impossible 15 years ago. Now, with the synchrotron, this can be done because structural biology become. So we have gathered a very, very excellent team to do that, and we have now can really do rapid co-crystallography, and this way devise new engineered molecules. And this is how it looks like. Part of the work is being done by a robot. So you take a protein, well, still at this stage people are working, and compounds and crystallization buffer, and then you feed a robot that makes mixtures of all different types of cocktails. And this is a rather robust robot, which later on takes these samples, put them also in incubator. At this stage, this is manual, this is done by a robot, and then it's being transferred to an imaging machine, which also says, oh, these are interesting crystals to look at, and here people come again, scale it up, run to the synchrotron, and solve the structure. Now, synchrotron is about 10 minutes drive, so this can be done. So it became a really engineering high throughput process. Just to show you how this really works. So this is all the crystal structure of the protein kinases that have been shown in different times. Some of them we still didn't publish. And I'm just showing you here the work in protein kinases because it's really linked to cancer. So this is, now that you really know how protein kinases look like, so all these blue entities are these weak inhibitors that wind to the catalytic core. So look, for example, what is the current run rate? Current run rate is 450 co-crystal structures annually. So they're solving about co-crystals, 1.5 co-crystal structure per day. So this really is a very impressive engineering task, and this way you can really develop drugs. And I'll show you a few examples. So this is now an example of a new scaffold. These are totally new, so this is advantageous because you can pattern them. They're small. So this is how it co-crystallizes together with tyrosine kinase PIK2, which we discovered, and with ABL. And you can see that it really binds slightly different to the two. And this shows you that when you superimpose them, they're here in red. You see, they're all small molecules. This is really good because you can really build up specificity and affinity in a very robust way. And they all can be superimposed with the ATP, with the adenine moiety of the nucleotide. So this is really going to the ATP binding pocket. So this is now showing you how this works. This is not a model. This is really real data. So this is the ATP binding pocket, positively charged regions, negatively charged region. So let's see how the first structure looks like. This is a scaffold. It binds with very weak affinity. So now you can see that you don't need to do sophisticated medicinal chemistry because you have the limitation of space and charge here. You have limitations. So for example, you can add another entity here and increase the affinity to micromolar. And this is not a model. These are all crystal structure, and these are all data. And you can add another entity, and you can get nanomolar. So very quickly, in a few months, you can get very potent inhibitors. And this is just an example here of how this can be used for deesterases, in which we have shown 4,000-fold increase in potency in a period of several months. So, for example, this is the first compound. This is the affinity and this is the structure. You add a benzene here, you increase the affinity significantly, you add another nitro here, you can see how these are all, nothing here is modeled, it's all real crystal structures. So now I'll show you an example of a new drug that will go into phase 1 very soon. As you know, there is RAF, which is a component of the signaling pathways that John described to you. There is a certain mutation in one of the isoforms of BRAF, which is a valine that becomes glutamic acid. It confers constitutive activation of RAF. It was shown to be prevalent in 66% of metastatic melanomas, in 36% of thyroid carcinomas, and in 10% of colon cancers. RAF is also a target for angiogenesis, as many angiogenic factors activate RAF. And so this is how it really works. This is the signaling pathway, which I didn't have time to elaborate. Receptor tyrosine kinase and the major information flow all the way from the surface to the nucleus. This is where RAF lies. In these tumors, this is activated, so it's constitutively activating the signal, and it loses the independent to be activated by ligand. And this shows you the prognosis of patients which have this mutation. This is metastatic melanomas patient with activating BRAF mutation. You can see that their survival is really much more grim as those which do not have the mutation. And it's even more severe in colon cancers. This is wild-type RAF, and this is the mutant RAF. So this is the number of patients discovered per year. It's still an orphan cancer indication because it's less than 200,000 patients, melanomas, thyroid cancers, and colorectal cancer. Remember, these are not all colon cancers. These are only those which exhibit this mutation. These should be the people that should be treated with this drug, not those which do not have the mutation. So this is using this approach that I showed you. We found an extremely new potent inhibitor of selective for BRAF. This is a very interesting approach because essentially it does not interact with the normal allele. The affinity is very significant difference. And the window is about, this is showing you the affinity versus of one of the inhibitors to the mutant RAF versus, so it's about 14-fold, and this one is 7-fold. So this, you can really dose it in a window of concentration to have basically not to touch the normal allele. It works very well in xenograft melanomas, and it shows you how these different drugs which block the growth of them. These are rather aggressive melanomas. And this is also colon cancer cells. So you can see these are some other RAF inhibitors, previous one, and this one you can find a dose which really is extremely efficacious. So summary. So what I've shown you today is a brief historical account of the fundamental concept of receptor tyrosine kinase, the signaling by growth factors, everything which led to where we are today from the 60s to the 90s. I showed you that this is a surprising result, which we just recently published in Molecular Cell, showing the different stage in which receptors are activated. I thought this is an important finding that people should be aware of, that receptor tyrosine kinases are activated in a two-state model because of the ordered reaction. I showed you the discovery of a tyrosine kinase inhibitor as a novel drug, which we think is going to change the outcome of some very severe diseases. And finally, I showed you new engines for generating of many new drugs. By the way, the RAF drug is now in final preparation, and it will be launched to a phase 1 clinical trial in the next few months. So this is some of the people who participated. The work in Plexicon is done in Berkeley here. There's some people in my work. Since one of the fathers here, you see, you should see your son. You know your audience. He's a postdoc in my lab out here. And this just shows you the entrance to Yale University Medical School, which is rather very pretty, and the historical library of the medical school, which is one of the nicest libraries in North America. Just for the entertainment, since we're in the movie business, some dancing tyrosine kinases. We'll finish. Thank you very much for your attention.
Thank you very much, Professor Schlesinger, for a brilliant talk. We still have time for in a few minutes, and I'll take questions for both Dr. Mandelson and Dr. Schlesinger, if there are any. Yes, please. Wait, so he can hear you.
If there is any connection using the AGFR receptor inhibitors, you set up a chemotherapy, we start with the big repair, and you also reverse resistance to chemotherapy. These two effects are interchangeable.
So the question is there a mechanistic explanation for why giving Erbitux with chemotherapy works? I have two answers for that. There's no direct mechanism that I know of, and I think it'll be different for each drug. But after chemotherapy, many of the drugs work by damaging the DNA, and we know that repair is reduced. So that's one explanation. But the other is, what's the purpose of chemotherapy? Today, we don't think of it as just killing the cell by poisoning the DNA. We think that it is probably working by inducing apoptosis through various mechanisms. And I believe that by combining 2 or 3 different agents, one of them might lower BCL2, another one might raise BAX, another might potentiate one of the other enzyme pathways. We get together a certain threshold that kicks the cell into apoptosis. And I think that those 2 mechanisms, and the third one, angiogenesis. And we're learning more that even chemotherapy can block angiogenesis too. Most of these oral agents work in the test tube to arrest cell growth, not to kill them, but in the human and the animals, it works better at killing, and I think that may be due to the angiogenesis. So there are many still possible explanations.
Any other questions? Comments? If not, thank you very much again.